Now that we've touched on all of the major players in the specific immune system, what I thought I would do in this video is kind of do a summary so it all fits together a little bit. So the first person or character we got exposed to was the B cell, which I'll always do in blue. So the B cell right like that. And what made that interesting is that every B cell has its own specific, or they have membrane-bound antibodies. But for each B cell, the membrane-bound antibodies on each specific B cell had its own variable portion. So this B cell, it'll be variable right like that, variable like that. And if I were to draw another B cell right here, I would draw the variable portion a little bit different. And this is why different actual B cells will respond to different antigens or different pathogens that have entered our system. And a B cell gets activated. It needs to get, let me write all this down actually. So if we're talking about a B cell to get activated, so let's talk about what happens when it gets activated or what needs to happen. So activation. It needs binding of the pathogen onto one of these membrane-bound antibodies. So binding of pathogen, or maybe we call it immunogen or antigen. Antigen. But that's not all. I mean, sometimes that's all you need, but usually you also need to be stimulated by T cell. So helper T cell stimulation. And you might say, where does the helper T cell stimulate this guy? Well, B cells were also antigen-presenting cells, so he'll suck this guy in, break him out, and present him on an MHC2 complex. So that's an MHC. Let me do it someplace where I can draw a T cell coming in. Well, let's say this is an MHC2 complex. This guy gets cut up. Part of him gets presented right here. And then an activated helper T cell. An activated helper T cell whose variable portion of their T cell receptor is specific to this could come along and activate this character. I'm not drawing that receptor well, but this is that right there is a helper T cell, and that is the B cell. Now, once it's activated, it starts differentiating, and it starts cloning itself, and it could either turn into effector cells. And this is true of B cells or T cells. Once they get activated, they keep cloning, and they either turn to effector cells or memory cells. Memory cells stick around a lot longer, so that in the future, you're going to have many more of this version of B cell. So if you get the same antigen or pathogen in the future, the likelihood of it bumping into this type of B cell is going to be higher, so the response will occur faster. The effector B cells they essentially turn into antibody making machines. So they'll say, gee, this antibody bonds to this antigen that we have in the system now. Let me just produce a ton of them. So it starts building up all of the cellular machinery, and it just starts producing antibodies like crazy. And I want to point out one thing that my wife pointed out to me when she overheard me making the last video. And she's a fellow in hematology, and a lot of hematology is immunology. So I definitely have to defer to her. She is the expert on this. In the last video, I kind of, very hand-wavingly said, oh, B cells, once they get activated, if they're the effector B cells, they produce antibodies. I want to be very clear. It is only the effector B cells that produce the antibodies, and the common term for them, if someone were to walk up to you and say, what cells in the body are producing antibodies, you wouldn't be wrong if you said effector B cells, but the common term that people expect to hear are plasma cells. Plasma cells. Plasma cells and effector B cells are the same thing. But normally when they say what happens to a B cell when it starts producing antibodies, they then call it a plasma cell. They don't call it a B cell anymore. And I want to make that very clear because my wife is like, well, I have attendings that if they asked me what cell in the body produces antibodies, and if you said B cell, they would say, no, wrong, it's a plasma cell. Or if you even said effector B cell, they wouldn't be happy. They wanted to hear plasma cell. This is the common term used in immunology and apparently rheumatology circles. Did I just say my wife is a hematologist? No, no, she's studying rheumatology. I get confused with all the ologies sometimes. Anyway, that's what the B cells do. And these antibodies can then go attach things and mess up viruses and antigen. Well, viruses are instances of antigens and bacteria and tag them for pickup by macrophages or other types of phagocytes. Those were the B cells. Then you have your T cells. And here I'm going to talk about T cells a little bit differently than I had in the last few videos, just to give a little bit more of a nuance. So there's two types of T cells. And you might be able to say, hey, there are helper T cells and cytotoxic T cells. And you're not wrong. But what I'm going to do is do a slightly different differentiation, just so that you are familiar with these terms. So there's two types of T cells, just like that. All T cells have T cell receptors. Let me draw the T cell receptors. T cell receptors just like that. But they also have these other proteins on them. And some of them have these proteins called, I'll draw it like that, called cd4 proteins. And some of them have, I'll do it in a different color, and some of them have what's called cd8. cd8. So this one right here would be called a cd8 positive T cell. It has the cd8 proteins on it. And this would be called a cd4 positive. cd4 positive T cell. I've never used these words before. You're like, gee, where are these coming from? Now, the cd4 receptor is the thing that wants to bind. This is the thing that helps to go to the MHC2 complexes. So most cd4 T cells are helper T cells. So this is, most of the time, and I want to make it very clear, immunology is this field, people are discovering things on a regular basis. So people are still understanding these things, and there's all sorts of special cases. But usually when people talk about cd4 positive T cells, they're talking about helper T cells. So this is normally going to be a helper T cell. Or you could call it a T helper, just like that. Likewise, the cd8 proteins, these are attracted to the MHC1 complex. These are attracted to MHC1. You can kind of say this is what brings them to the cells that have the cancer, that have expressed antigens on their MHC1 complex. So most of the time, cd8 positive T cells are cytotoxic. So cytotoxic T cell. And oftentimes, before a cell gets activated, they just describe it as a cd4 T cell or a cd8 T cell. And then after it becomes, for example, in a cytotoxic T cell, after it becomes activated and starts wanting to kill things, then maybe you call it cytotoxic. But this is all, wordplay. I think you get the general idea. But just to remember what they do, this guy, we just said, he wants to bond to the MHC complex. So you have MHC complex plus presenting some antigen. This is MHC1 right here. We learned in the last video, every nucleated cell in the body expresses an MHC1 complex. So this is a case where something wacky is happening inside this cell. Maybe a virus has infected it. Maybe it's cancerous. It needs to die. Otherwise, it's going to keep producing protein, or keep producing viruses if it's infected by a virus, or otherwise it's going to keep dividing if it's a cancer and infect the rest of the body. So the cd8 kills infected cells. I'll just say bad cells, because I don't know if you can, cancer really isn't an infection. Kills bad cells. Cells that are, if you don't kill them, they're going to keep producing viruses or keep splitting and spreading the cancer. While T cells, they're attracted to professional antigen presenting cells. So, and I always do a dendritic cell right here, because those are the best antigen presenting cells. And they have MH2 complexes, and it's digested some antigen and it presents it right there. And then that activates the helper T cell. And then when the helper T cell, all of these guys, once they're activated, they all go into they all start differentiating into effector and memory cells. An effector helper T cell, it does a couple of things. So when we're talking about helper T cell, it can activate It can activate B cells. And it also releases cytokines. Let's say this guy gets activated, he'll also start releasing these chemicals, which are really those alarm bells that tell other people to really get in gear. Maybe B cells and cytotoxic T cells start proliferating more rigorously. Actually, part of the T cell the cytotoxic T cell activation can be assisted or kind of be given a boost by these cytokines, so by these alarm bells. So this guy is the alarm ringer. Alarm, alarm. While the cd8 cells, or the cytotoxic T cells, when in their effector mode, they kill cells. They kill cells. And of course, in the memory mode, there's just a bunch of copies of these originals around that are ready and more than there originally were, so that in the future, if something like this or something like this occurs, they're going to be activated faster because they're going to be bumped into faster. So hopefully that clears up a little bit, and I introduced a little bit more terminology. But I really want to stress my wife's point, because she said, hey, you don't want people out there saying B cells produce antibodies, even though it is effector B cells. Once activated B cells that are differentiated into effector B cells, those are what are producing antibodies, because when they go to medical school, people are going to want to hear plasma cell.